Just, there we go. So remember that the electrons, as they move through the electron transport chain, cause what to happen? Why do the electrons going through the electron transport chain, why is that important? Adeline?
Well, it makes the waste product of water.
It makes the waste product of water. What's oxygen's role in this process? Olivia? It's the last acceptor, so it's what we call the final electron acceptor, and it is that because it's the most electronegative, right? And so therefore, why is it important that we get these electrons moving through the electron transport chain? What happens as the electrons move through the electron transport chain? Adeline?
I was going to say it pumps the hydrogen molecules in the inner membrane space.
That's right. It pumps the hydrogen into the inner membrane space, and so therefore we create this high concentration of H pluses in the inner membrane space. Can somebody else remind me why that's important? Why is it important to have a high concentration of H pluses in the inner membrane space? Megan? Yeah, right. It diffuses back into the matrix here through an protein, which is also an enzyme, ATP synthase, and when the H pluses, like this H plus diffuses back through from high to low concentration, what happens to the ATP synthase? Isabel? What do you think? The shape changes. So remember, for an enzyme, the shape, the part of the enzyme that's important to be in the right shape is the active site, right? And so therefore, in this case here, when it changes shape, it puts it into the right shape for the substrates to bind to the active site. What are the substrates for this enzyme to act on? What are the substrates? Megan? ADP and an inorganic phosphate, which means that it's just a phosphate not attached to anything. So ADP plus P with a sub I come in, they bond to the active site, and I'll put over here what we get out then is ATP. So therefore we make the ATP, and for every NADH then, when we talked about NADH, it pumps 3 H pluses through. So three H pluses then can diffuse back through. So one NADH makes how many ATP? Makes 3 ATP. How about FADH2? 2, right? One FADH2 makes 2 ATP. Why does FADH2 only make 2 ATP? Why do they only make 2 versus the 3, Isabel?
Because it enters the electron transport chain like later, so it only goes through 2 of the complexes.
That's right. FADH2 drops its electrons off. Did I write this on here yesterday? FADH2? Yes, okay. Drops its electrons off, bypassing that whole first complex. So therefore, its electrons only go through 2 of these protein complexes in the electron transport chain. Therefore, only pump 2 H pluses, so only 2 H pluses can diffuse back through the ATP synthase, making 2 ATP. All right, so did I have you guys total yesterday? All right, I did. Did I have you total the total for oxidative phosphorylation, how many total ATPs are made? I didn't think so. All right, so this is, I think, maybe where we left off at the end of the hour yesterday. Question?
I'm sorry, the 1 NADH and the 2 ATP, where is that coming from again?
Oh, all right, so NADH drops its 2 electrons off here and moves through the electron transport chain. When it does that, it pumps 1, 2, 3 H pluses. Each one of these H pluses go in, making 3 ATP.
Okay.
But because FADH doesn't go, their electrons doesn't go through this, there's only 2 H pluses and therefore less energy.
Okay.
All right, and less ATP. And actually, before we total this, let me ask you this. This pumping of the H pluses is active transport. Do you guys remember where the energy comes from to pump the H pluses from low to high concentration, because there's no ATP used in here, so it's not ATP energy. People always forget this part, like where the energy comes from. Olivia? Well, what about the electrons? It has to do with the electrons moving from molecule to molecule in the electron transport chain, but it's more specific about that movement of electrons. What?
So this one become oxidized, but when juice is going down like that, and that creates energy signals like transferring as it goes down.
That's right. We have a whole series of redox reactions here, reduction and oxidation. So why do these two electrons leave NADH in the first place to go to this first molecule in the electron transport chain? So I think I might have acted this out yesterday, all right? But if I'm the first molecule, the two electrons come to me from NADH. Why? Because I'm more electronegative than NADH, so therefore the electrons are drawn to me. I just became what? Oxidized or reduced? Reduced. The next one down the line, here, next one down the line, more electronegative than me, so the electrons leave me and go to the next one. Now I become oxidized, the next one becomes reduced, and so on and so forth. These redox reactions, as Isabella said, release energy. That energy is the energy to pump the H pluses, okay? And so what I'd like you to do is, using what we just talked about up here and not looking at anything else, I'd like you to total how many ATPs are made in this third step, oxidative phosphorylation. See if you can do it just based upon what we have on the board. Don't look at anything else, because it'll tell you in those packets if you get it right. So don't look. And see if you can reason through how you're going to figure that out. You can talk amongst yourselves if you'd like to help each other. So how do we figure this out? Madeline?
So I noticed that there's 4 NADHs coming from the original steps over on the left side.
So 2 here and 2 here.
So then you add those 2 NADHs to the 6 NADHs that come off the citric acid or Krebs cycle. So that makes 10 NADHs. So you multiply that by 3, because 3 hydrogen ions are being pumped through, and then they go through the ATP synthase to make the 3 ATPs. So that's 30 right there. And then for the 2 FADHs, they pump through the hydrogen ions. How many hydrogen ions do they pump?
2.
2. And then
Therefore,
they make
2
2
ATPs.
or 4 ATPs.
2 of these, but there's they each make 2, so 4 ATP. So therefore, total, we add these guys together, and so we can add to our picture that this process is 34 ATP, this third step. So therefore, our total is, for the whole process of cellular respiration, is 34 plus 4 gives you 38. All right. So the other 4 are coming from the 2 from the Krebs cycle and the 2 from the glycolysis. Does that make sense?
How does those 2
These 2?
Oh.
Yeah.
So that comes off.
But then you have to add the 2 ATPs.
Yeah, so glycolysis makes 2, net of 2. Krebs cycle makes 2. Oxidative phosphorylation makes 34, so we get our total of 38. And last night in the notes, I talked about how they roughly estimated between 36 and 38, that it's not an exact science here. So this is rough for our intents and purposes, this is fine. But remember that I talked in the notes about NADH can't get across the membrane, so this NADH doesn't literally carry its electrons to the electron transport chain. What it does is gives its electrons to electron carriers already in the mitochondria. So if this gives its electrons to an NAD+, it becomes NADH and goes to the electron transport chain. But if it gives its electrons to the NAD or FAD, it becomes FADH2, and you make a little bit less energy, and so that's why there's those fluctuations there. All right, and so that's where the totals come from. So, so let's go back to the oxidative phosphorylation packet here and just go over the rest of the answers. So we were, remember here where we were at. I'm going to leave this picture up. Did we start going over these? Yes. All right. I have no recollection where we left off. So for this part here, circling it, remember, is the part that we have the inner and outer membrane here. So this is the inner, outer membrane. This right here is the inner membrane space, and this part right here is the matrix. And so that's what we're looking at there. All right. So then, do we go over number 2? Yes. Anybody have any idea?
I think we started this. I think we started.
I didn't think we started it either. Okay.
No, we didn't
All right.
start it.
Okay. All right. All right. So that's number 1. All right. Does that make sense? Number 2. Highest concentration of H+ ions you should have is in the inner membrane space. B. How do the hydrogen ions reach this area? They are pumped through the transport proteins in the electron transport chain. All right, pumped meaning going from low to high concentration. 3, why is energy required to move the hydrogen ions across the membrane? I just answered that because they're going against their concentration gradient from low to high. 4, high potential energy electrons provide the energy necessary to pump hydrogen ions across the membrane. A asks you what molecules carry these high potential energy electrons. These are NADH and FADH2. They carry the electrons to the electron transport chain. Where do these electron acceptors come from? NADH is made in glycolysis, the link reaction, and the Krebs cycle. FADH2 is only made in the Krebs cycle or citric acid cycle. C, when the electrons are released from these molecules, what else is produced? And that is the hydrogen ion. So remember that when NADH gives off its electrons, that H loses an electron and becomes an H+. The same thing with FADH2. So it's the hydrogen ions or H+. 4D is the release of an electron from one of these electron acceptors, oxidation or reduction. It's losing electrons, so you should have that that's oxidation. 5A, what molecule is the final electron acceptor? That's your O2, your oxygen. B, what compound is formed as a final product? You should have water. 6 is any ATP produced in the electron transport chain? You should have no. And 7 is any ATP used, and you should have no. All right, so that's just an electron transport chain. So then we move on to this picture. This is the chemiosmosis part. So again, going back up to this big picture on the board, oxidative phosphorylation has these 2 parts, the electron transport chain, and this part is chemiosmosis. So this occurs first so that this can occur second. The whole thing with those 2 together are collectively called oxidative phosphorylation. So what we just looked at in the packet was the electron transport chain. This picture now we're looking at the chemiosmosis part where the H+ are going to go back in to make your ATP. All right, and so they're all collectively called oxidative phosphorylation. So when we look at this chemiosmosis part, the H+ are now going to diffuse back in. And so number 8 says describe the movement of these H+ ions through the membrane. And so they go from the inner membrane space to the matrix from high to low concentration, so therefore it's diffusion, and they go through the protein called ATP synthase. So then number 9 asks you, would free energy be required for the hydrogen ions to move in this direction? And you should have no, there's no energy required to do this because it's moving down the concentration gradient. 10, what is the name of the protein that's moving through? ATP synthase. 11, the flow of ions through the protein channel provide free energy to do work. And what process requires energy? And that is the making of ATP. So to make ATP, to put the phosphate back onto ADP, to make your ATP, this is an endergonic reaction. So this needs energy, and this enzyme, remember enzymes lower the amount of energy needed for a reaction to occur so it can happen faster. So therefore, what's happening is ADP and the phosphate are the substrates. The ATP synthase changes shape, and the ADP and the phosphate, remember what enzymes do in their active site. So the ADP and the phosphate come in and they orient themselves, and as the ATP synthase does the induced fit, like hugging it, puts the ADP and the phosphate together and puts them in the right spot where they can bond together, and therefore lowers the energy that's needed for this reaction to happen. And so let's look at number 12 on the other side here. During oxidative phosphorylation, what molecule is being phosphorylated? You should have ADP. 13, under ideal conditions, each NADH results in 3 ATPs, FADH, and 2 ATPs. What's the total number? So you should have, that's what we just did on the board. So there were 10 NADHs times 3 gives you 30 ATP for NADH. And there were 2 FADHs, 2 multiplied by 2 gives you 4, so you get a total of 34 there. 14, considering all stages of cellular respiration, all of them, how many ATP molecules are produced from one glucose? So I'll just put these answers up here. So, as we wrote on the board, glycolysis is 2 ATP, no ATP in the link reaction. Krebs cycle is 2 ATP. Oxidative phosphorylation is 34 ATP. We add them all together, we get 38. Okay? 15 asks you, because of its role in aerobic respiration, aerobic is in the presence of oxygen. Oxygen is essential for most living things on Earth. It says describe the role of molecular oxygen in aerobic respiration. So they have this little paragraph here, but we've already talked about this, that oxygen, its role is to be that final electron acceptor. It's the most electronegative, so it draws the electrons through, which then allows the H pluses to be pumped into the inner membrane space so that you can make your ATP. So that's the role of the oxygen. The role of the oxygen is not to make water. Water is just a byproduct. The role of the oxygen is to draw those electrons through, okay, so that it would be the most electronegative molecule. Alrighty, so 16, you had to fill in this chart. So this chart here is a nice little review thing that you should be able to do off the top of your head. So let's look at here. So it has your reactants and your products. So reactant number 1, glucose. What phase is it used? It's used in glycolysis, and that location is in the cytoplasm. For oxygen, oxygen is not used to oxidative phosphorylation, and that location is in the mitochondrial matrix. A lot of people put for this the electron transport chain, but if you look on the board, where oxygen is actually used is on the other side of at the end of the electron transport chain, so it's not actually part of it, it's in the matrix there. Then the product, CO2, where is CO2 produced? In the link reaction in the Krebs cycle. And location is in the matrix there. Both of those occur in the matrix. Water is produced only in oxidative phosphorylation. And that is, the water says mitochondrial matrix in the inner membrane. But really, it's one of those electrons, the inner membrane, it's not really part of the electron transport chain. So the water is actually produced, I would just say, more realistically in the matrix. And then lastly, you get your 38 ATP at the end. So where are those made? There were 2 of them in glycolysis, 2 of them in the Krebs cycle. This should not have ATC. and then 34 of them as a result of oxidative phosphorylation. So where do these occur? Glycolysis occurs in the cytoplasm. Krebs is in mitochondrial matrix. Oxidative phosphorylation is in the inner membrane as well as the matrix there. So that's that. Alrighty. So 17 asked you about substrate level and oxidative phosphorylation. There's two different ways that ATP are made in this reaction of cellular respiration. Oxidative phosphorylation here, and these two, glycolysis and the citric acid cycle, are through substrate level phosphorylation. Both of them, you're adding a phosphate onto ADP to make your ATP, but the phosphate comes from different sources. So in glycolysis and the citric acid cycle, where these phosphates come from to make your ATP is they take them off of a molecule. So they call it a substrate. So it takes it off of a molecule and puts it on to ADP to make your ATP. Same thing with the Krebs cycle. To make your ATP, they take a phosphate off of a molecule and put it onto ADP. That's different than in oxidative phosphorylation. Oxidative phosphorylation, the phosphate is not attached to anything, so it's free floating, and that's the only difference between them. So that's the answer there for 17. 18 is an application question where it's asking you about the higher pH. Where would you find the higher pH? So you have to think back to pH. Higher pH, the number is larger. Higher pH, is that acidic or basic? Basic, right? Bases, do they have more or less, or what happens to the hydrogen ion concentration? Hydrogen ion concentration decreases. Acids give off H pluses, so the higher concentration of H pluses makes it more acidic. So if we look here at the board and we look at the matrix versus the inner membrane, the matrix has a lower concentration of H pluses, so this inside is more basic than the inner membrane space, okay, because the higher concentration mass. All right, so then what are we at? Number 19. So 19, you have this enzyme, hexokinase, uses ATP to transfer a phosphate to glucose to form fructose diphosphate. Suppose that the cell only has glucose available for energy and that the activity of this enzyme is suddenly stopped. So look what's going to happen in the cell. So it says that this enzyme uses ATP to transfer phosphate to glucose. So what part of cellular respiration are we talking about here? This is this first part, glycolysis, but not only in glycolysis, the very first part of glycolysis, because it says it's using ATP. This is the energy investment phase. And so there's this enzyme that catalyzes one of these first reactions by phosphorylating the molecules. And they're saying here, if this enzyme stops working for some reason, suddenly stopped, what's going to happen in the cell, or what's not going to happen in the cell? Cellular respiration, we're not going to get past, we're never going to make the pyruvate, which means that none of the rest of cellular respiration happens. So this cell is not going to be able to make any ATP. It's going to likely die, all right, is what's going to happen to that cell. Okay, so then the last one, 20. Is this the last one, 20, or is there one more?
20 is two parts.
There's two parts. So we have prokaryotic cells. They also need energy. They need to make ATP, yet they don't have any mitochondria. What's the only process that doesn't occur in the mitochondria? Glycolysis, right? So therefore, that's the answer to A. Then B asks you where, it tells you that the link reaction and Krebs cycle occur in the cytoplasm of the prokaryotic cells, but you need a concentration gradient across the membrane just like we have in the mitochondria, so that ATP can be made as the H pluses diffuse through. What happens, do you think, with prokaryotic cells? Anybody have an idea, a hypothesis about what happens with prokaryotic cells with this? Prokaryotic cells, what are we talking about? What is bacteria, right? So let's just talk about just the general gist of the bacteria. Bacteria don't have any organelles. They do have a cell wall. I'm just going to CW for cell wall. Underneath it, they have a cell membrane. No organelles. They do have ribosomes, so I'm going to put some dots here, and then they have a big circular chromosome, so I'm just going to put C-zone for chromosome, that's their DNA, this is their ribosome. So this is what a bacteria looks like. So basically, they're saying that glycolysis, the link reaction, and the citric acid cycle all take place in the cytoplasm here, because they don't have a mitochondria. So all of that's going on here. So, but they still need to do oxidative phosphorylation, which requires the pumping of H pluses so that you have a concentration gradient. Where do you suppose that the electron transport chain is located in a bacteria cell? Jennifer?
The cell membrane.
Yeah, it's located in the cell membrane. If we look here, if I put these three here as the big protein complexes in the electron transport chain, their electron transport chains are in the cell membrane. And so then what happens is then they pump their H pluses in the space between the cell membrane and the cell wall. So then they also have ATP synthase right here, all right? And these H pluses can diffuse them back in, and they can still make ATP to be used. Does that make sense? All right, and so therefore they are also able to make ATP without a mitochondria. Okay, and go through all three stages. All right, so that is cellular respiration in a nutshell. Okay, so good practice would be to see if you can, you should be able to sit down with a piece of paper and you should be able to do this, all right, if you really know it. All right, so you should be able to go, okay, glycolysis, where does it occur? I'm going to go through what happens, what's the energy investment phase, what's the energy payoff phase, how many ATPs are made, where does it go, okay? So that's a good practice there. So what I'm going to do is next show you a video, it's just a short little video that now that you have a good idea of cellular respiration, this will make it more meaningful for you. This video, let me just show you where. Alright, I'm on the website that goes with our textbook. Remember that there's a drop-down menu. You can go to it as practice tests and practice quizzes, animations, and so on. If you click over here, just so you can see where I'm going, there's a menu here that says BioFlix. These are little videos on some of the topics. Not all chapters have them, but there's one in cellular respiration, which we're going to look at. Let me just, again, show you with these BioFlix here. You can go through a slideshow. I'm going to do an animation. I'm clicking on the bottom here where it says animation. There's a tutorial where it'll ask you questions. I don't know why it's blinking like this. It usually does not do this. There's a study sheet that you can use and print off, like this is a good practice, like the stages of cellular respiration, where does it occur, what molecules go in, what molecules go out, can I summarize what they are, and so on, and what happens in them. Let me go back, oh I lost my BioFlix. Right. So that's aha, that's not what I want to do. Don't do this. And then there's a little quiz I wanted to show you. So then you can take a little quiz and see if you understand it, and so on. So that's available to you in a little cellular respiration thing. So let's go back to this. Go back to the animation. Alright, it should be playing.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process.
I want to point out with the citric acid cycle that it's a cycle because you start out with your product at the end is what you originally start out with, and we draw it like this to show that it's a cycle. But if you notice in there, it's just in the mitochondrial membrane, and these reactions occur, but it's not like they sit and go like this in a circle, the cycle. All right, it's just random in the mitochondria, but you just end up with the last reaction with the first molecule that you started out with.
Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondria. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy.
The redox reactions.
The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
That always startles me that stat, 10 million ATP molecules per second. That's a lot of ATP. Considering one glucose, you get 38 ATP. So you got a lot of cellular respiration going on, alright, to generate that. So it's very quick and happens very fast in a lot of it. Alright. Alright, so last night we wrapped up the notes and I have a couple of questions that I'm going to have you talk with people around you to see if you can answer off the top of your head. So last night I introduced, in addition to cellular respiration, this process of fermentation. So can you tell what the purpose of fermentation is? Can you describe when and what happens in fermentation in us? And then number 3, I learned last hour that people didn't know what I was talking about with the carbon cycle. It says, how does cellular respiration relate to the carbon cycle? You guys ever learned the carbon cycle? Some people say yes, some people say no. Do you remember learning about the water cycle? Yeah. And how water is kind of cycled throughout, like goes through living things, goes into the atmosphere and so on. So the carbon cycle is the element carbon atoms, how they move from the atmosphere into living things and back into the atmosphere. So I'm going to have you see if you can relate cellular respiration and the carbon and the molecules of cellular respiration, how it relates to carbon moving from atmosphere to living things back to atmosphere. So if you can do something, challenge you to think a little bit. So these 3 questions, I'm going to give you a few minutes to do that right now. Okay? So see if you can do it. See if you can do it off the top of your head. And if you understand it and somebody else doesn't, you can let them know. Yeah.
Well, it has something to do with
I know what, okay.
There's a lot of pressure. You get about a minute or less per question.
All right, you guys. So let me ask you this. What part of cellular respiration can occur with or without oxygen? Michael. Glycolysis. So glycolysis is the only thing that can occur if no oxygen is present. If there's no oxygen, pyruvate doesn't enter the mitochondria, which means none of the rest of this happens. So only glycolysis can occur without oxygen. So glycolysis is coupled with fermentation. So fermentation, with there's no oxygen, if only glycolysis can occur, you can only break down glucose to the 2-pyruvate and only get 2 ATPs per glucose molecule. So that's it, and that's the end. And so then to make more ATP, you need another glucose molecule to be broken down, and so on and so forth. And so, can somebody explain to me, I said that glycolysis is coupled with fermentation when there's no oxygen. What does fermentation do? What's the purpose of it? Yeah, it recycles the NAD pluses here. So just to reiterate that, what I talked about in the notes, what happens is normally if oxygen's present, these NADHs are made. These NADHs carry 2 electrons, so I'm the NADH, I carry the 2 electrons, and I drop them off and get rid of them in the electron transport chain. I become NAD plus again, I come over here, get 2 more electrons, I become NADH, drop them off, I become NAD plus. All right, you get the picture? And so therefore, NAD plus and NADH are constantly being recycled in the cell. But what happens is if there's no oxygen, that NADH has the 2 electrons, this isn't happening over here, so the electrons stay with it. It never gives the electrons up, so it stays forever as NADH. And therefore, what happens is, as your cell is just going through this process, all your NAD pluses are going to be converted to NADH, and none of your NADH are dropping off the electrons. So they all stay as NADH, and your cell is going to run out of NAD plus. If it runs out of NAD plus, can it do anything with glucose? No, it can't even make 2 ATP molecules, and you don't get any ATP and no energy whatsoever. And so what the purpose of fermentation is fermentation does this. It converts NADH back to NAD plus so that you can continue to go through glycolysis and you never run out of NAD plus. So that's the purpose of it. Now in this process, instead of the pyruvate becoming acetyl-CoA and doing the rest of the process of cellular respiration with no oxygen, the pyruvate, in order to convert the NADH to NAD+, the pyruvate is converted into one of 2 different things. Either ethanol, if it's like yeast, or lactic acid if it's us, all right? So the purpose of fermentation is not to produce alcohol or lactic acid. Those are byproducts. In the production of taking pyruvate and making alcohol or lactic acid, you regenerate your NAD pluses, and therefore glycolysis can continue. So which one of the 2 types of fermentation for number 2 occurs in us? Lactic acid fermentation. Can somebody tell me what's going on there? So we have, and so we do this in our muscle cells when there's no oxygen. So the pyruvate, instead of going into the mitochondria and doing the rest of it, gets converted to lactic acid. And in that conversion of pyruvate to lactic acid, that's how we regenerate the NAD pluses so that we can continue to go through glycolysis. Can somebody explain to me, when lactic acid fermentation is occurring in our muscles, how do we create that cell having an anaerobic condition even though you're still breathing? So you as an organism are still breathing. Megan?
If you eat like more ATP faster than your oxygen can go to those muscle cells, then you need lactic acid fermentation.
That's correct. All right, so you have to be using a muscle cell and going through cellular respiration at a very fast rate to generate ATP. Remember though, to do this last step, you need oxygen to be the final electron acceptor. So you can create a situation where you are using a muscle so much, all right, so if I'm lifting weights and doing it as fast as I can for as long as I can, I'm going to get a muscle cramp in here. That's the lactic acid. So what's happening is I am breathing, I'm taking in oxygen into my body. The oxygen goes into my blood. The blood carries it all throughout the body, including to my muscles that I'm working. But the oxygen's getting there, but not enough oxygen is getting there. And so what happens is I can't breathe fast enough to get oxygen into the cells to do cellular respiration as a fast enough rate that I'm using it. So then when you're not getting enough oxygen, your muscle cells convert to doing just glycolysis because it can't do the rest of cellular respiration. And so then it starts doing fermentation to regenerate that NAD+. The byproduct of that is lactic acid. Guess what lactic acid is? It builds up in your muscle cells, create pain. That's actually a good thing because the pain will trigger you to stop, all right? And therefore, it allows you to breathe and the oxygen to get there, and so then you can do the rest of cellular respiration and your muscle cells stay alive and they don't die for overworking and creating the anaerobic situation. All right, so I'm going to run out of time here. So carbon cycle, anybody have any ideas about that? Cellular respiration and the carbon cycle. Grayson?
We breathe out CO2 and it goes into the atmosphere, and then the plants take in the CO2 and create glucose, and then either us or other animals that we eat will eat the glucose and then eat the carbon and then become breathe out.
That's correct. All right. Did everybody follow that? Yeah. All right. We'll stop. We'll stop. So you guys,